Hi everyone, my name is Matt. I'm the Manager of Application Development and Analysis. Thank you all for joining me today for my discussion on democratizing NMR. And so I hope that by the end of my presentation, you'll understand what I mean when I'm talking about the democratization of this technique. And I'm going to try my best to highlight a few recent projects we've been working on to support these ideas. And so namely, I'll touch upon the validation of some industrially relevant qNMR assays with benchtop NMR spectroscopy. And so for those of you who aren't familiar with our company, and I'll go into this in a little bit more detail, but Nanalysis manufactures benchtop NMR spectrometers, and we've been a pioneering force in this area for over 10 years. And so I'm very excited to be sharing some of our recent work with you today, so let's get started. So I'd like to very quickly go over what I'll be presenting today, just to give you an idea of what to expect, because it will be kind of split up into several different sections. So I'll start by discussing both the geographic and end-user distributions of NMR as it currently stands, which will lead me into briefly touching upon the concepts of underserved users and the proliferation of NMR. So after introducing our permanent magnet technology, I'll move into a bit of a primer on quantitative NMR, or qNMR as it's commonly referred to, and I'll elaborate on some things that are quite important to consider when you're using this technique. So I also want to talk a little bit about the current status of NMR in industry, so basically who uses it and for what applications. And I'll try to present a path towards accessibility for this technique. And then finally, I'll show you a few examples of some work we've done on hydroxypropyl beta dex, some poloxamers, as well as some polyoxyl ethers. And I'll try to kind of provide an outlook for the work we're continuing to perform, so we're sort of an outlook towards future projects. So Nanalysis is a global leader in magnetic resonance research, and we've been growing globally and steadily since our foundation in 2009. We offer direct sales in several countries, including the United States, Canada, and Germany, and we have a network of over 45 dealers across the whole world. So we're based in Calgary, Alberta, in Canada, and we manufacture everything here as well in-house. And so in 2020, we purchased RS2D in France, who have really helped us increase our offerings in the magnetic resonance space, specifically within the MRI sort of technology. And in 2021, we also purchased One Moon Scientific, which are allowing us to greatly enhance our software capabilities as well. And at the end of last year, sort of leading into the beginning of this year, so into 2022, we also added K-Prime Technologies to the team, another Calgary-based and owned company. And their sales and service experience will help us greatly enhance our customer experiences. So a lot of growth on the horizon. Now, when we think of NMR and discuss sort of its timeline of innovations, things go way back, like a lot of scientific instrumentation. Some key leaps and bounds and technological advancements have really allowed us to access the powerful high field systems that we have today. And they've really enabled groundbreaking research and truly made NMR an indispensable technique for, honestly, anyone performing any kind of exploratory or synthetic chemistry of any kind. It's really hard to envision doing any kind of chemical research without having at least some access to NMR. And so while it's tempting to think of benchtop NMR technology as a new technology, and I guess this is really true when you look at it in the grand scheme of all NMR history, it really has been around for over 12 years or so now. So since 2010, year over year, we've noticed a pretty significant increase in not only the number of people who have heard of benchtop NMR technology, but have also thought of many new ways of using it. So it's very exciting and rewarding from our perspective to have people kind of reach out to us with new ideas. And we've seen that a lot, and it's becoming more and more common year over year. So some of the increased visibility that benchtop NMR has sort of enjoyed is definitely due to its potential to bring NMR to what are termed underserved users, but it's also part of a bigger picture and sort of a newer picture, honestly, and that's kind of what I want to get into today, of NMR being used as an analytical technique in industrial QA/QC. And this is something that I think we need to have more conversations about in terms of expanding the scope of NMR testing in industry to reflect the fact that currently with benchtop NMR technology, we do have unparalleled access to this technique. We've never had this level of accessibility, and we definitely expect this accessibility to continue to grow over the coming years. And so when we take a look at the geographic distribution of high-field NMR instruments, we quickly noticed that 8 or so countries alone own approximately 80% of all of these instruments worldwide. So obviously, this is a problem. There are major accessibility concerns that need to be addressed. These accessibility concerns are typically just not a problem with a lot of other traditional analytical instruments. So it just isn't really feasible for many industries to invest in a high field instrument. It has both, these high field instruments always have very large upfront and recurring costs, and this is especially problematic in the sense that many countries who are so important to global manufacturing don't have access to a technique that's this powerful and useful. So the problem here is really that we have a mixture of underserved users, as I've mentioned a few slides ago, and this is typically caused by accessibility issues. But we also have underserved markets, so again, such as industrial QA/QC. And so as it currently stands, you can see from this end user distribution of NMR spectroscopy that the majority of NMR use continues to be reserved for research and exploratory purposes. So namely, roughly 76% or so of end users continue to use NMR for qualitative or structural elucidation purposes. This doesn't leave a lot of room for industrial quality control. A major part of this is due to the fact that these industries have not really had access to NMR for the last 60 plus years, like they have with a lot of other affordable and less space-consuming instruments and techniques like gas or liquid chromatography. So historically, GC was developed in the early 60s, and LC followed suit in the sort of mid to late 60s. And by the time the 80s rolled around, both of these techniques were widely used in various industries. And it's important to mention that they weren't just used for research purposes, but also they were used for quality control significantly. And so scientists across the world have now been using these techniques, like GC and LC, across various industries for over 40 years. This is really important to keep in mind. They've had over 40 years to not only perfect the instruments and the techniques, but more importantly, they've had over 40 years to build up a library of standard test methods using these instruments. So until benchtop NMR started to really kind of ramp up in accessibility in the early 2010s, NMR just simply wasn't this accessible. It did not approach the accessibility of techniques like GC and LC, like with other analytical techniques. And so the primary purchasers of this technology, benchtop NMR technology, were initially academic institutions for undergraduate lab teaching. But the market has expanded significantly to include many different industries. And now we're really starting to see what was likely happening with GC and LC in the 80s, where end users are trying to find new ways of using this benchtop NMR technology. So the development isn't just coming from benchtop NMR manufacturing companies now, it's also coming from the customers. And so this is kind of what I mean by the need to democratize this technique, not only geographically, but also to underserved users. And also from an application perspective into industrial QA/QC spaces to help kind of balance the scales between using NMR for qualitative purposes and more into quality control. So I've been mentioning underserved users and the proliferation of NMR a bit since I've started the presentation. So I kind of wanted to take a little bit of time to discuss these concepts in a bit more detail. So when we speak of underserved users, we're typically referring to researchers who can't, or honestly just don't want to, outsource their NMR analyses. These users typically don't have access to NMR on site, and most of them honestly can't imagine purchasing and maintaining a high field system. So with regards to the proliferation of NMR, we can and should, at both from a geographical proliferation perspective and from an application and industry proliferation perspective, we should really focus on this a little bit more because we know that there's an appetite for access to NMR across various industries because people reach out to us all the time. So this isn't just coming from us. We honestly get requests all the time for people in industry asking how they can use benchtop NMR. So we need to continue providing access to this technique outside of the more traditional qualitative and structural elucidation spaces. And so this presentation today, if there's kind of a takeaway for people listening in, is this presentation is really part of an effort to try to expand the use of NMR across various industries, which is something that we're really focused on. And so the miniaturization of benchtop NMR technology by analysis was done sort of in two ways. One, by using rare earth permanent magnets, so specifically neodymium iron boron, and arranging them in a highly homogeneous and compact array, which has really allowed us to use minimal amounts of magnetic materials and to have very limited stray fields, which means the instruments are safe to be around. So if you think back to your high field instrumentation use, there were certain things such as your phone and your credit card and things like that that you couldn't bring near the instrument. This is not a problem at all with these benchtop NMRs. So you can get as close as you want with your phone, your credit card, your USB drives. It doesn't matter. So they're quite safe to be around. They also don't require cryogens or any significant maintenance, and non-experts can use them quite easily, as I hope to kind of show you today. And secondly, the miniaturization of the electronics means that we've been able to condense these down. So if you think about when you think about a high field instrument, you're not only thinking about the magnet itself, but also the consoles that go with it. So we've been able to condense those into small circuit board stacks, and we've also embedded a computer so that spectra can be processed directly on the instrument if desired. Of course, you can also use a separate computer to do this if you prefer. So Analysis currently offers two main product lines. We have our 60 megahertz and our 100 megahertz instruments. And as I mentioned on the previous slide, these are based on neodymium iron boron magnets, so both models are. And we have a 1H19F only model, so we term this the E model, for customers doing more routine analyses. And we also have some more complex heteronuclear experiments that are available to users hoping to collect additional data from our instruments. So these would be the Pro instruments. All the data I'll be presenting today was collected on our 60 Pro instrument line, but I will only be showing proton data. So now that you know a bit more about the instrument we offer, I kind of want to situate them within the context of current technologies. So when we think of traditional high-field spectrometers, we know they offer incredible resolution and sensitivity. This is true. But they do have very low accessibility, and it's just not feasible for labs to incorporate them into their workflows, whether this be due to price, size, or the requirement to have expert staff on site at all times to maintain them. At the other end of the spectrum, we have low field and low resolution relaxometers, which are much less expensive, but can only be used for relatively basic applications. So relaxometry is extremely useful, but it doesn't work for all applications because it is low resolution. We kind of like to think of benchtop NMR as bridging the gap between these two, meaning that by addressing the accessibility concerns that are inherent to high field instruments, but still providing high resolution analyses. So there are many underserved markets that could certainly take advantage of the strengths of NMR as a technique, but the combination of the price, the size requirements, and the expert staff for maintenance is just too much for a lot of industries to handle, meaning that NMR honestly just kind of tends to fly under the radar. We often hear from people in industry who say, oh, I loved using NMR in grad school. There's no way we can afford one here in our industry. So no matter how powerful of a technique it is, if you can't have access to it, then it kind of affects it. So I think most of you will also have heard about the helium shortages that are becoming more and more common. I doubt that these are likely to be resolved anytime soon, and I doubt that we're going to be addressing this in any significant way. So this is something else to keep in mind when thinking about precious high-field NMR technology. So switching gears a little bit, I'd like to very quickly go over some of the key concepts that are relevant to quantitative NMR, or qNMR, as you'll often see it referred to. So one of the most unique and useful things about NMR is that it's inherently quantitative, which means that the integration area of each individual signal is directly proportional to the number of chemically equivalent nuclei that are giving rise to that signal. So this is important because it means that a specific analytical standard of the target analyte is not required. It's really important to note that the integration areas are only quantitative if the acquisition parameters are properly set. So when we think about HPLC, for example, the instrument needs to be calibrated, and you need to carefully consider how to properly separate all of the components during a chromatography run. With NMR, we don't need to separate anything. So the components of interest are all in solution, and we essentially obtain a snapshot of everything in that solution at once. Also useful with NMR, these analyses are all non-destructive, so if you do have a valuable sample, you can simply recover it after analysis. So if we kind of situate ourselves within a QA/QC setting of some kind, regardless of which one it is, then we know that accuracy is paramount. I mentioned that these acquisition parameters are important for this, so I kind of want to walk through this together to kind of show you what I mean. So The spectrum on the right that you're seeing now was collected as part of a sample experiment that I was preparing last year. And I think it serves as a great example of this. So in this spectrum, I used a fairly standard scan delay of just 1 second, so I didn't really optimize anything. And if you look at the integrations under the peaks, you'll notice that if we normalize the peak for the two methyl groups to 6, so that signal at 3.39 ppm, we've normalized that to 6. Then the integrations of the other regions don't give us the expected values. So based on the chemical structure, all 4 of those regions should all integrate to 1. So these are all related to a single proton each, so these should all be 1. But they're all underrepresented in this case, and this is because our scan delay is too short. So our interscan delay does not give these spins sufficient time to relax between pulses. So we're underrepresenting these integrations. So what do we do? Well, typically when quantitative NMR data is required, the T1 times for the signals of interest will be determined. Then a user will take the longest of these values to set the scan delay. So typically a value between 5 to 7 times that longest T1 will be used to ensure that all of the spins of interest can be considered fully relaxed between pulses. So our instrument allows users to quickly and easily perform T1 experiments, which enables them to then in turn determine the appropriate scan delay to use for their experiments. So for the molecule in the previous slide, the longest T1 time was found to be approximately 4.1 seconds, which I've highlighted in the red box here, meaning that we should be aiming for a scan delay of approximately 20.5 seconds, which is 5 times longer. So if we set our scan delay to 20 seconds, so you can see the acquisition parameters on the left, as I had them initially set up. So if we set our scan delay to approximately 20 seconds, the few additional seconds that we get from the active scan time will ensure that we're certainly above the value of 20.5 seconds. So if we collect another spectrum using the same acquisition parameters, and the only thing we vary is the scan delay, then we can immediately see the impact that this has on the integration areas. So in the spectrum on the right, the values are much closer to those we would expect, and they conform perfectly to the structure shown above. So this is an example of why optimizing your acquisition parameters is important for accurate and quantitative NMR data. It's really not a complicated process, and our instrument really makes it straightforward, but it needs to be done whenever a new assay is developed. So you can see here that if you're doing exploratory work and you're not sure if you've synthesized a specific molecule of interest, the spectrum on the right with the optimized scan delay will give you much more confidence that you did compared to the one on the left. So as more people start to use this method, it's important to realize that there are a few other key things to consider when you're preparing for a qNMR analysis. So it's extremely important to be accurate at every step of the way, which means using proper weighing techniques. And so we've found that, at least from our experience, weighing errors tend to be one of, if not the most common sources of error in qNMR. So this is extremely important. On top of this, proper liquid dispensing methods need to be used, which means either using a volumetric flask or a calibrated micropipette. This is important. It's also crucial that your acquisition parameters are optimized for collecting qNMR data. So in a simplified way, this basically means that you need to use an adequate number of points to sample your data. And you also need to set your repetition time and your spectral width to enable optimal results. This can be overstated. So typical acquisition parameters, let's call them the default acquisition parameters, used to collect traditional one-dimensional NMR data will almost never provide accurate quantitative results. And related to these acquisition parameters, scan delays must be chosen to ensure that all spins of interest are fully relaxed between pulses. So we just discussed this in the previous example, but it bears repeating again because it is extremely important. So benchtop NMR technology, as I mentioned, since 2010 has been thriving, and we would cite many reasons for this, but I kind of want to highlight method development, technological innovation, and also application validation as some key reasons why this field will continue to evolve. And so today, I really want to focus on this last point, where I will do my best to try and provide you with examples of how benchtop NMR can be used to address specific industry problems by providing market-driven analytical solutions. Now, let's talk a little bit more about the current uses of NMR in industry. And so for decades, the bulk of QA/QC work in industry has been reserved for other instruments, such as HPLC, FTIR, GC, IC, mass spec, to name just a few. There are many instruments that are used in these QA/QC spaces. And I've said this already, but you'll hear me repeat it again. High-field instruments for NMR are expensive to purchase and maintain, and they do require expert staff for maintenance. They also necessitate dedicated rooms because they are quite large. On top of this, they do require liquid helium and liquid nitrogen to cool the superconducting magnets. Don't get me wrong, I love high-field NMR. Everybody on my team loves high-field NMR. So these instruments are fantastic tools, and it's great to have to actually climb a set of stairs to insert your sample into an instrument, something HPLC people might not understand, but not every single application requires the superior peak dispersion and sensitivity that traditional high-field instruments provide. So for many industries, using the minimum viable product is a crucial way of cutting operating costs. And for some applications, it's overkill to use high-field spectrometers. So for certain applications, the minimum viable product might not be a 600 megahertz spectrometer, it might be a 60 megahertz spectrometer. So to highlight this, I've included a stacked plot of a phosphorylated Kraft softwood lignin on the right. And this came out of one of our previous projects. And all I really wanted to show, you don't need to know anything about lignin. All I wanted to show was how similar the phosphorus NMR spectrum, so 31P NMR, of our sample collected on a 60 megahertz instrument was compared to the one collected on a 400 megahertz instrument. So you can see a bit of additional fine structure in the aliphatic region and also in the carboxylic acid region at 400 megahertz, but really the quantitative information that we extracted from both spectra was almost identical. So the respective integration regions are almost exactly the same. So in this case, if you are thinking about sort of routine quality control analysis of something like lignin, there really isn't a huge reason or a huge push to go to a 400 megahertz spectrometer if you can extract the same quantitative data at 60 megahertz. Now, it's clear that benchtop technology is poised to provide a path towards accessibility for NMR in industry, especially in these QA/QC settings. And we can point to many industries that could certainly benefit from this. Just to name a few, pharmaceuticals, food testing, lignin analysis, polymers, rubbers, there's so many more. But the very first steps required to do this properly is to demonstrate what's termed equivalency to existing standard methods that already require NMR. So evidently in the future, we would love to develop numerous new methods that make use of NMR across various industries. But these take time to develop, and there needs to be an established need for specific assays from industry. So to start, we thought it would be more appropriate to find published standard methods that people already use, which do require high-field NMR, and demonstrate that we can perform these analyses at the benchtop level. So once we've done this, our plan is to reach out to various agencies, such as the United States Pharmacopoeia, the European Pharmacopoeia, the Japanese Pharmacopoeia, ASTM, AOAC, ISO, there's many more, to discuss the possibility of modifying the wording in their published methods so that the ones that are amenable to benchtop NMR spectroscopy can be written as such. So we understand that many industries require these methods and rely on these methods, but we often get questions like, okay, this is great, but can you perform this analysis according to monograph method whichever in the USP? And even if we can, and we know we can, and we've demonstrated we can, if the official published method states that you need a 300 megahertz or stronger instrument, then that's kind of what needs to be used. And so today, I want to focus on 4 different United States Pharmacopoeia and European Pharmacopoeia monographs, and I will demonstrate equivalency to them using only our 60 Pro instrument. So all of these analyses are very straightforward, they're easy to set up, and they really require minimal amounts of processing. So before I get into that, I really just want to mention one last thing. And I just want to mention that there really is a space and a desire for NMR in more industries. So there is a significant need to address these accessibility concerns, and this is what we really mean when we talk about the democratization of NMR. So having this technique available on the bench can really decrease reliance on outsourcing analysis and it frees up high field instrument time for more demanding analyses. So these high field instruments are crucial to certain areas of research because of their incredible sensitivity and resolution. But many analyses simply don't require these. So we need to free up these instruments for the analyses that do require them. So finally, just a really quick comment to mention that these benchtop instruments are highly customizable, so they can be tailored to accommodate a wide variety of heteronuclear experiments, which can feature nuclei such as lithium, boron, carbon, fluorine, phosphorus, and others. So we've already been quite involved in providing access to NMR to various industries that either had very limited access to this technique in the past or just no access at all. So I'm showing a few projects we've worked on within this context. So hydrogen content determination in crude oils, the investigation of H2S scavenger activity, which was published by one of our customers at Brenntag. We've also looked at some battery electrolytes via proton and fluorine NMR, and we've done a lot of work with the phosphorus analysis of lignin, which I mentioned a little bit earlier. We've also developed a method for the determination of lithium content in brine pools via lithium-7 NMR. So we've presented all of this work previously in various forums, so please feel free to look these up or just contact us if you'd like more information. I've included a few references below to some relevant peer-reviewed publications that we've put together on these topics. So please check them out if you're interested. And I guess at this point, I've rambled on long enough. Let's get into some actual NMR. So first I want to mention that all the work I'll be presenting was developed as part of an effort to demonstrate equivalency to the United States pharmacopoeia monograph methods and also the European pharmacopoeia monograph methods. And so these monographs are the standards for most assays performed in the pharmaceutical industry. And they serve as a great starting point for us to really start branching out into this QA and QC space. So today we'll be specifically targeting larger molecules, so more along the lines of oligosaccharides and polymeric materials. And the work we're showing here is based on some monograph methods that already make use of high field NMR. So I really need to point out how rare this is before we start talking about this in more detail, because there are only about a dozen or so methods that explicitly require the use of NMR in these pharmacopoeias. And I need you to really think about this for a little bit, because when I looked into it, I counted well over 5,000 monographs that were published in the United States Pharmacopoeia alone, and a little over 10 of them use NMR. So while the work I'll show here establishes equivalency to these NMR methods using a benchtop system, we're also trying to target replacement of certain methods as well. So hopefully I'll be able to present some of the work that we're doing in this specific area to you down the line at some point. So let's start by discussing hydroxypropyl beta-dex. So cyclodextrins are ingredients in many approved medicines, and a big reason for that is because their interiors are hydrophobic and their exteriors are hydrophilic, which allows them to form complexes with hydrophobic compounds. So if you think back to host-guest chemistry. And so this means that they can essentially encapsulate molecules within their central cavity. This has led them to be used for tailoring drug delivery and even to mask undesired smells. So if you think of Febreze or even dryer sheets, which bind this fragrance in the cyclodextrin and release it when it's heated. So as a really brief introduction, there are three major forms of cyclodextrins. And the only big difference between them is how many glucose subunits they contain. So it's pretty simple. Alpha cyclodextrin contains 6 glucose subunits. Beta cyclodextrin has 7, and gamma cyclodextrin has 8. That's it. So on the right, we've got a beta cyclodextrin because it's built up of 7 glucose subunits. So you can count them there if you want. And very simply, cyclodextrins are prepared from starch via breakdown from some enzyme. And this enzymatic breakdown process is kind of what sets various companies apart. So companies that manufacture these cyclodextrins. So hydroxypropyl beta-dex can often be referred to by its full name, so 2-hydroxypropyl beta-cyclodextrin. And it's a form of beta-cyclodextrin that's undergone a transformation. So by reacting the sort of native, if you will, beta-cyclodextrin with propylene oxide, some of the OH groups in the molecule will be transformed into hydroxypropyl groups, hence the name hydroxypropyl beta-dex or hydroxypropyl beta-cyclodextrin. So one of the tests that's defined within both the United States and European Pharmacopoeia monographs for hydroxypropyl beta-dex is called the molar substitution test. And it's used to determine the average number of hydroxypropyl groups per anhydroglucose unit. So the analysis is pretty straightforward. What we're doing is comparing the 3 protons on the methyl group in the hydroxypropyl fragment. So here I've labeled it as A1. To the C1 glycosidic proton, so A2 in this figure. So because this A2 proton won't react with the propylene oxide, it kind of acts as our sort of constant, so to speak, against which we can compare the methyl peaks. So the official method in both monographs state that a spectrometer operating at a frequency of at least 250 megahertz is required. But our analyses show that our 60 megahertz instrument demonstrates sufficient performance metrics in both SNR and peak resolution. So it does the job extremely well. So what we've done is we've analyzed 6 different hydroxypropyl beta-dex samples from various suppliers. So for each product, we prepared 3 individual samples, and we ran each of these in triplicate for a total of 9 analyses per individual product. Then we compared the molar substitution values that we obtained to those provided by the suppliers when that information was available. So on the right, you'll see a typical proton spectrum of hydroxypropyl beta-dex on our 60 megahertz instrument. So you'll notice how straightforward this analysis is, since both the A1 and A2 regions are very well separated. So the quantitative analysis takes less than 5 minutes, and the sample preparation really just involves weighing approximately 10 milligrams or more of a solid, which is the hydroxypropyl beta-dex, in a vial, and dissolving it in D2O. That's it. That's all you need to do. So I want to walk you through a bit of a summary of our results, and I know this table seems like a lot, but I promise you I'll walk you through it. So here we're comparing the results we obtained at both 60 megahertz and also 400 megahertz. So the same physical samples were used for the analyses at both fields. So the MS values under the product name, so in the leftmost column, refer to the values that are reported by the supplier. Second column from the left, so samples 1, 2, and 3, refer to the individual physical samples that we prepared. And the values reported in the A2 columns are actually the integration regions of interest, and these are the average of triplicate runs with the RSD values shown in parentheses. So in all cases, the A1 region was normalized to 100, so I'm not showing it here. Finally, the last column on the right shows the molar substitution values that we obtained from our analyses, and because they matched exactly at both 60 megahertz and 400 megahertz in all cases, I've just included them as a single column here. So you'll see that for every sample, our values match the label values exactly, except for the one that I've named HPB-H107, so the third one down. So while we do know that the other samples, so 1380, 1460, and W7HP, we know that the manufacturers determined these molar substitution values using NMR. The supplier for the H107 sample wouldn't provide us with the method they used to determine this value, so we really don't know if it was done using a validated approach or how this was determined. As such, we feel pretty confident in the fact that our calculations matched exactly at both 60 megahertz and 400 megahertz for our work. Excitingly, we've also started developing an analyzer interface for this hydroxypropyl beta-dex analysis, which we believe to be a good candidate for this. So our vision for this is one where users would be provided with some, let's call them standard tubes. So these are just tubes that contain pure hydroxypropyl beta-dex samples with a very well-known molar substitution value. So the user could periodically calibrate the instrument using these tubes as prompted by the interface. So once the interface has determined that sufficient time has elapsed between calibration, it would simply ask for a specific tube. Then it would enable the software to adjust both the phase and baseline to ensure that accurate integrations are obtained. Then the customer could simply analyze their quote-unquote unknown sample or their new sample by simply inserting their tube in the instrument, pressing go, and then waiting for the molar substitution value to be displayed directly on the screen. So this type of analyzer interface isn't unique to this assay. We ourselves have had success with this approach previously, so for determining alcohol content in kerosene. And ideally, these types of analyzers would really allow for a more high-throughput QA/QC analysis from non-expert users, which is not something that's traditionally done with NMR. So with this approach, a user really doesn't need to be familiar with processing or interpreting NMR data. As long as they can prepare a sample, which, as I've mentioned earlier, is quite simple, and follow the prompts on the screen, they can obtain an accurate QA/QC result. So moving on a little bit, I want to discuss some of the work we've done on poloxamers. And again, these are assays that are outlined in both the United States and European pharmacopoeia monograph methods. So we will specifically be looking at a test method termed the weight percent oxyethylene test. But let's dive into a tiny bit of background beforehand. So poloxamers are block copolymers, which, as the name implies, are made up of individual polymeric blocks. So in this case, there's a central polyoxypropylene chain, and this one is hydrophobic, which I've highlighted on the figure on the right. But there are also two flanking polyoxyethylene chains, which are hydrophilic. So the length of each chain can actually be adjusted during polymerization. So this means that a wide variety of structurally similar but very chemically distinct products can be obtained. So even if you're not familiar with these polymers at all, it's pretty easy to see right away how unique these polymers can be, considering that they contain both a hydrophilic and a hydrophobic fragment, and that you can tailor them to fulfill specific chemical property requirements. So because of their unique properties, poloxamers have found widespread use as surfactants, emulsifiers, drug carriers. They're honestly everywhere, and they're present in so many products we use on a daily basis. But because the properties of these polymers depend heavily on the exact sizes and lengths of these polyoxypropylene and polyoxyethylene chains, the quality control of these products is crucial from an industrial perspective. Okay, so what does NMR have to do with all of this? Well, proton NMR can be used to accurately determine the weight percent of oxyethylene in these poloxamers. So as described in the monograph methods, the 3 protons of the central polyoxypropylene methyl group will be compared to the combined integrations of the methylene protons and the proton of the central polyoxypropylene methine group. This is a mouthful. On the next slide, everything is labeled along with the NMR spectrum, so it'll make a lot more sense. So it's actually really useful that all regions of interest can be easily distinguished at 60 megahertz. So this analysis really is perfectly suited for benchtop technology. So the sample preparation involves only dissolving about 100 or 200 microliters of sample in D2O with 1% DSS as an internal chemical shift reference. And then you transfer that solution to an NMR tube. That's it. So I really don't want to diminish the work that we've done, but it really doesn't get simpler than this. So we analyzed 8 different poloxamers from different suppliers, and we've compared the results to those to the expected values based on the specific polymer. So before I get into the results, I kind of want to mention that these poloxamers have a widely used nomenclature, and I guess I'll call it the P nomenclature, which allows us to know exactly what weight of oxyethylene we expect for each sample. So very briefly, so if you look at the name of one of these poloxamers, let's do P124. So the first 2 numbers multiplied by 100, so 12, will give us the approximate molecular mass of the polyoxypropylene core. And the last number, so 4 in this case, multiplied by 100 will give us the, multiplied by 10, sorry, will give us the approximate polyoxyethylene content in weight percent. So this is the value that we're after in this test. So for example, P237, 7 multiplied by 10, we're expecting a value of approximately 78%. So below, I'm showing a proton spectrum of a typical poloxamer on our 60 megahertz instrument. And here I've highlighted and labeled these A1 and A2 regions to make it easier to see what we're integrating. And so in the table on the right, the results of the analyses at both 60 megahertz and 400 megahertz are shown. And like with the hydroxypropyl beta-dex in the previous example, 3 individual samples of each poloxamer were prepared, and each sample was analyzed in triplicate at both fields. So the weight percent oxyethylene values are shown here with the RSD values in brackets again. And the expected values for each poloxamer is shown on the right, and you'll notice that some of them are presented as a range. So the ones that are presented as a range are the specific ones that are included in the published monograph methods. So those ranges are dictated by the United States and European pharmacopoeias. The other poloxamers are not included in these monographs, but we studied them regardless because there are so many of these poloxamers available. We wanted to test as many of them as possible. So as you can see, the values that we obtained at 60 megahertz match those from the 400 megahertz analyses extremely closely, and the RSD values are very small. So this indicates that this is a very reliable and effective analysis at the benchtop level. Again, I really want to highlight, this analysis takes about 20 seconds once you've prepared your sample, so it's really quick. So finally, I want to move on to the final application, which is actually split into 2 separate USP, so United States Pharmacopoeia monograph methods. And we'll be looking at 2 different polyoxyl ethers, and the structures are shown here, and I'll elaborate on these structures in a second. So polyoxyl ethers, unsurprisingly, are polymers, as the name kind of implies, and all of them contain polyoxyethylene fragments. They're widely used as surfactants and emulsifiers in soaps, deodorants, creams, detergents, moisturizers, conditioners, exfoliants, sunscreens. Basically, most of us have almost certainly come into contact with one or multiple polyoxyl ethers today. So because they're used in so many consumer products, like with the hydroxypropyl beta-dex and the poloxamers, the quality control of these is crucial. So the first polyoxyl ether we will focus on is polyoxyl 10 oleoyl ether, and I've shown it below here on the left. So in addition to that polyoxyethylene fragment, it also contains an oleoyl fragment. The second polyoxyl ether we'll focus on, so the one on the bottom right, is the polyoxyl 20 cetosterol ether. And so in addition to the polyoxyethylene fragment, this one also contains a cetosterol fragment, which is actually a mixture or an average of cetearyl alcohol and stearyl alcohol-based fragments, which are, to put it in more chemically friendly terms, C16 and C18 chains. So in both cases, we will use proton NMR to determine the number of repeating polyoxyethylene fragments, so the subscript N value in this case. And this will be done according to the average polymer length test described by the monograph method. So for polyoxyl 10 oleoyl ether, the protons of the oleoyl fragment, so A1 in this case, minus those of the alkene fragment and those directly adjacent to the ether oxygen. So A1 will really be the protons highlighted in this green box in the figure. And so we will compare these to the integration of the polyoxyethylene protons, so A2 in this case. It's really simpler to just describe this using a figure. So if you look at the one on the right, I've highlighted the A1 protons in green, and these are in that region between approximately 0.8 and 2 ppm. The A2 protons do overlap with the terminal hydroxyl proton, so the blue one, and the protons adjacent to the ether oxygen, so the red one. But these are subtracted during the calculations. So in the spectrum, you'll see that the alkene protons don't overlap with any of the regions of interest, so we just won't consider those. So like with the poloxamers, each analysis only takes about 20 seconds, and the sample preparation is really straightforward. So these are actually waxy solids, so all you need to do is melt it at approximately 60 degrees, and then you add a certain amount of this melted solid to a deuterated chloroform solution containing 1% tetramethylsilane as an internal chemical shift reference. Then you transfer it to an NMR tube, and then you run your analysis. So the table below shows the results of our analyses at both 60 and 400 megahertz, where, once again, we prepared 3 physical samples for each product and analyzed each of them in triplicate. So for this study, we looked at 3 products from different suppliers. And in 2 cases, our values match those from the high field nearly exactly. Or in all cases, they match the high field data exactly, but in 2 cases, they match the expected values. And so you'll notice that for the second sample, the average polymer length actually comes in just under 10. And so we want the value to be between 9.1 and 10.9 according to the USP monograph method. And this one actually comes in at 9.0 at 60 megahertz and 8.9 at 400 megahertz. So we thought that was interesting. For the polyoxyl 20 cetosterol ether, the method of analysis is essentially the exact same. So once again, you melt your waxy solid. This one's a little bit easier to melt, so you heat it to 50 degrees, and you prepare the sample in exactly the same way. In this case, we actually expect a value between 7.2 and 25.0 for the average polymer length. And so here, the protons of the cetosterol fragment, so A1 in this figure, are compared to the combined integrations of the polyoxyethylene protons, so A2. So if you take a look at the figure on the right, I've highlighted the regions of interest again, and similarly to the previous example, you'll see that A2 does overlap with that hydroxyl proton and also the CH2 next to the ether. Those are subtracted during the calculations again, so they aren't a problem. Here again, we analyzed 3 different products from different suppliers, and the values we obtained are exactly what we would expect, which is very encouraging. So the 60 megahertz data once again matches the 400 megahertz data almost exactly, and this is another analysis that takes approximately 20 seconds per run. And in this case, even though we have a broader range of acceptable values for those, the number of repeating units between 7.2 and 25.0, they're almost all exactly around the 20.0 range. So you might remember this slide from earlier in the presentation, but I thought it would be a good time to show it again. So like I mentioned previously, we've already made headway when it comes to incorporating NMR into a variety of different industries. For very different analyses, but there's still a lot of work to be done here. So ultimately, this will end up depending on what various industries need. So we really do encourage you to please reach out to us if you have a problem that you think NMR could help with, because we're always looking for new applications. We've already added several new analyses relevant to industrial quality control just from today's presentation alone, but we will keep going. So by targeting these industrially relevant assays, we know that we can keep expanding on the uses for benchtop technology across new industries, both by taking advantage of analyses that don't require high field instruments, but also by replacing other methods that could benefit from the ease of use of NMR, which is something we're actively working on. So I know that I've already provided you with a ton of information this morning, but just to close, I want to go back to the hydroxypropyl beta-dex analysis that we showcased earlier to mention that we'll be publishing a white paper on this topic later this year. So specifically, we aim to demonstrate method verification via both an intra and inter-laboratory study, and we want to touch upon everything: precision, accuracy, robustness, selectivity, and a variety of other parameters. And I also want to mention that studies on additional cyclodextrin derivatives are underway, and we will be demonstrating equivalency to HPLC methods that are used for assays of other published cyclodextrin monographs. So some of these will be purity determination using qNMR with an internal calibrant, but others will be molar substitution tests, which for some reason in these monographs are dictated as used with HPLC. So all the work that I presented today was possible because of a mixture of us at Nanalysis doing our own research into these topics, but also because of industrial collaborators reaching out to us with questions or comments about the work that they really want to do with benchtop NMR. And so we always do our best to try and figure out what various industries need, but realistically, nobody can tell us what an industry needs better than the actual industry. So please, developing these applications takes time, but if you believe that benchtop NMR could help you, please let me know. Reach out to us. We'd be happy to chat. So finally, to conclude, I really hope that I've been able to convince you all that benchtop NMR can provide opportunities to democratize the use of NMR across various industries. And specifically, we firmly believe that QA/QC protocols can take advantage of the small footprint and low cost of this technology. So especially for analyses that just flat out do not require high field instruments. we've been able to demonstrate equivalency to industrially relevant assays, all following published and official United States and European pharmacopoeia monograph methods. And specifically, we looked at assays for hydroxypropyl beta-dex, poloxamers, and polyoxyl ethers. This is really just the beginning for us. So my primary focus is to continue performing these types of analyses according to different methods, including more from these pharmacopoeias, but also from other agencies such as the ASTM, AOAC, ISO, and many others. So I know that this is the beginning of what will certainly be a long process, but it's essential for us if we want to continue offering benchtop NMR technology to underserved markets. And so ideally, we would do this by also developing additional analyzer interfaces, like we've been working on with the hydroxypropyl beta-dex, to really reduce the expertise requirements necessary to operate an NMR instrument. So I'd like to very quickly thank a few people, but namely two of my colleagues at Analysis, so Susie and Juan, who've been extremely helpful in developing this work. And I would like to thank all of you today for joining me and for listening to me ramble on about NMR and some of the work we've been doing in this benchtop space. So you're more than welcome to reach out to us at any time, and I would be more than happy to answer as many of your questions as I possibly can.